So thanks again for joining us here today. We are in session 2 of the 2022 PhysiCell workshop and hackathon, and working on our first dive into PhysiCell, learning about the structure of the projects and how to work with them. I'd also like to point out that all of the materials for this talk are at this GitHub link here, or at this QR code where you can download the slides and the sample code. And later on today, we will upload this recording onto YouTube and post a link to that as well. Our goals today, we're going to go kind of a refresher and understanding the sample and template projects built into PhysiCell. We're going to go over the structural projects, remind ourselves about cells, their behavioral phenotype and cell definitions, and start learning about the different workflows that we use to build PhysiCell sessions, models, starting with today in this session with the basic modeling workflow. And then in session 4, we'll introduce the intermediate workflow. And tomorrow we'll get into the full workflow. For the basic workflow, we're going to learn to populate, build, and run a basic model and visualize the data in Python. So some key background here. First of all, you have to remember that there are sample projects or pre-built projects that are bundled with every PhysiCell download. So hopefully you've been able to download and run PhysiCell. Probably is in a GitHub directory, if you're like most of us. And there are several key rules in the make file. So you download PhysiCell and go into the root directory. And the first thing that you'll see here, so I'm going to go and clean up here. First of all, just clear my terminal window. Make reset. So make sure I'm all cleaned up. But you type make list projects, and you will see a list of all the built-in projects in PhysiCell. So now what you can do is you can choose any one of those and populate the name that particular project. So if I, for example, type make worm sample, that will take all the source code files for that particular project and put them where they belong. And then the second time around, you can type make to compile that project. So I'm going to go ahead and just cancel that. We'll make it later. And suppose now you want to switch between projects. What you can do is type make reset, and that will get you back to a clean starting point. And then you could do make list projects again and see what projects are available. So there are a number of projects that are available in PhysiCell. The one you'll be using the most is called template, the template project, which is like a basic starting point to build a 2D or 3D project. And so that's often where you're going to start any new project. And so now if you go into PhysiCell and look at the structure of the directory, so I'm just going to open my root directory here. If you look through PhysiCell, you'll see that there are a broad variety of folders here. Of course, the most important place is the root directory. That's where your main executable is going to go. There are other directories like add-ons and beta testing and the diffusion solver. But the directories that are most important for you in your day-to-day work will be the root directory, where the main source goes, main.cpp, your make file, and then the executable you make when you compile will be there. The config directory, so we can kind of go in here, is where your settings file will be. And by default, that will be an XML file called PhysiCell settings. The other important directory is custom modules. That's where we're going to put our custom code. And if you populate out the template project, then there will already be two files waiting for you there, custom.h and custom.cpp. And those are the files you would typically be modifying for your project. And then lastly, output is where your data are generally, by default, your data will be stored when you run your project. So almost all of our work on a day-to-day basis will be in these red directories. So let's learn a little bit more now about some of the key entities that are in PhysiCell. First of all, each cell agent is, like an agent-based modeling framework based in biology, the cell really is the key thing here. And so we have cell agents, which each of which have a unique integer type, and also a character, like a human readable string name of the type name. Say, oh, I'm like a PhysiCell, like I'm a fibroblast, or I'm a CD8 positive T cell, or I'm an epithelial cell. So based on the cell definition, when you created that cell type, it will have an integer, unique identity, integer code for what type of cell it is. So you can do quick and easy comparisons, and also a character string name, so you can kind of keep yourself sane, and also unique integer ID. Each cell will keep track of its own position and its velocity. It will have a state, which will have a few variables that are really important for particular interaction testing, and then a behavioral phenotype, including intracellular model and data included there. So the phenotype really is what you're going to learn about in session 3 next, really groups things by behavioral types: cycling information, death information, secretion uptake information, motility, mechanics. All that will be kind of grouped hierarchically by topic in the phenotype. And then lastly, something called custom data, which is where we're going to store any custom data we might need for our own biological models that are above and beyond the built-in things. And each cell has its own built-in methods to keep track of cell division, cell death, change of type. Say if you want to mutate from a cancer cell to a mutant cell, or differentiate from a stem to a differentiated cell. We have ways to easily access and sample the microenvironment, the ways to secrete things into the environment, ability to find other cells, built-in mechanics by potential functions, of course, motility, so random birth and motion, and now more recently, ways to ingest other cells by phagocytosis, to damage other cells by like an effector attack, or even to fuse with other cells. And so we'll be going through those behaviors in particular in sessions 3 and 6 and 7. So ways to access some of the key information in PhysiCell. First realize that each cell agent is a member of the cell class. And then they have these data, so the type name is that human readable string I was telling you about a moment ago. Here's our integer type and an integer ID. So these are really easy to access. There is a vector called position to really understand and see at a very quick glance where the cell is in space. It's really important that you never write to the position vector, because if you do, it'll mess up some kind of the internal data structures that help the broader simulation package keep track of where the cells are. Instead, what you want to do is there is a function called update position. It can use to manually write that will properly update the cell and all the appropriate data structures. But for simplicity, you can certainly read it. And then there's another vector called velocity to say which direction we're moving. And then you have the cell state as an object, the phenotype, custom data, and then a list of functions that act upon the cell. And we'll be talking about those a lot more tomorrow. So it's kind of where this is all going. Each cell has an instance of the cell state called state, which we'll talk about in a moment. In particular, there's a vector of cell pointers called attached cells, and he used the methods attached cell and detached cells to easily add or remove cells from that list. And we'll talk a bit more about that tomorrow and Wednesday. Another important structure is called, there's a vector of cell pointers called neighbors. And this will be a list of all cells that are within mechanical interaction distance. And you can use that for custom functions. And one thing that's very nice is that this data structure is automatically updated by the mechanics built into PhysiCell. So you never have to look for this, it's already there. And then there's something else built into the state called simple pressure, which you use for some really basic mechanical transduction. The phenotype, which we'll get into more in the next session, again, hierarchically organizes all the key cell behaviors, things like cycle information, death information, which would be not only like the rates of entering different death cascades, either apoptotic death and necrotic death, but then once you're in those death cascades, the parameters that govern how they're regulated, about how long it takes to conclude apoptotic death, for example, how quickly you lose volume and shed it into the domain. And then, so all these aspects are grouped by different aspects of cell behavior. The core behaviors are all already implemented and then hopefully debugged over the years. So you don't need to roll your own and worry about how you divide a cell in half and instantiate a new daughter cell, where you place it. All that stuff is built in. So you can focus on then is programming the phenotype in the cells. Say, what are my behavioral rules, and how do these behavioral rules change the core processes in the cells? How do I vary my birth rate? How do I vary my death rate? How do I vary my secretion rates? Rather than having to write those really low-level aspects, those have been implemented so that you can use them without worrying about them and really focus on your biology. Cell definition then is like a template for a cell. That it's a class that basically says, here's my cell of a certain type, and here are my default parameter values. So how's the phenotype already set with specific values, and then a list of functions that are assigned to that particular cell type. So if you have particular behavioral hypotheses for a fibroblast, you would have a fibroblast function attached to the fibroblast cell definition. And so the nice thing then is once you have a cell definition or few, you can find the cell definition and create new cells that already copy over all that stuff to say, I've got a fully fledged fibroblast that's ready to go and instantiated over in this position. So we'll be using those quite a bit in some of the projects tomorrow in particular. But even today, now because we have the graphical model builder, it's easier and easier and easier to create a new cell definition and set up at least these default parameters graphically without having to do all this stuff in C++. So let's just take a moment to think about the modeling workflows. And let's see, I want to make sure that I'm staying on time here. Yeah, we're doing pretty well. So there are a variety of workflows you can kind of think about. They start from very basic, and today in this session, we're going to introduce the most basic modeling workflow. The idea that you would take somebody else's existing project or sample project here today, change some parameter values, compile it, and run and visualize the data. So that's the most basic workflow, which I think is already a great place to start if you're working in a team. Then later today, and by session 4 in particular, we'll introduce the intermediate workflow. So imagine now you would start with a template project. You would go ahead and compile and build that, so you're ready to go. Then we would go into a graphical model builder and start defining the key components of your simulation, the diffusible substrates, the different cell definitions and their default parameter values. And actually what you'll see is we have now a built-in studio option where you can run and visualize the results right there in the graphical model builder. And so by the intermediate session, you can build fairly sophisticated, intermediate level models that have even basic interactions like predation without having to write any C++. So this gives you a really strong start to say, I've got my environment. So I kind of say where do my cells live? I got all the different players ready to go. I have the different cell definitions, and I can set the parameter values and do this hopefully in a very, very consistent and error-free way. And then when it's time to do the full modeling workflow, that's where you say, now that I've built up, my initial starting point, now I'm just going to worry about writing functions that act upon the cell behavioral parameters. And I don't have to do all this setup stuff in C++, which would be lots and lots and lots of code. So we want to really avoid that. So this cascade of workflows allows you to really kind of get started. And then when you're ready to do more, to add the dynamics of the phenotype, then you go into the full workflow and write a few C++ functions. So that'll be what we do tomorrow. So let's look at this basic workflow. So what we're going to do here today is we're going to take a built-in project and do some minor changes to parameters. So we're going to populate and build a project, edit the settings, run, and then view the results. And so just like we showed you here a moment, I'm going to open up a terminal window here in my PhysiCell directory. Now, my PhysiCell directory is just in GitHub. So you can kind of see I'm in my home directory, GitHub, PhysiCell. This works on Windows too, by the way. And so I'm going to type make list projects and see what I have. And I'm going to populate the heterogeneity sample. So I'm going to type make. So see right here, my list of projects, I have heterogeneity sample. So make heterogeneity sample. And it's going to see I was copying a bunch of source files to kind of where they belong in that project structure. So this only moves the files around. It didn't actually compile anything. So we need to type make one more time to actually compile and build that project. So we'll give that just a moment. And hopefully you've been through your setup tutorials, so this is no problem. And I'm already compiled, it looks like. So next step is we are going to go ahead and start editing the configuration files. I'm going to show you how to do it kind of old school, but it's just a kind of a typical code editor. And I think then Namika and the others in sessions 3 and 4 will introduce the graphical model builder for the first time to edit these settings. So I'm just going into config. And depending upon your operating system, if you're in Windows, you just kind of type the name and it'll open it up with whatever is associated with your file. In Macklin, I'm going to type open and then the name of my file, PhysiCell settings.xml. And that'll open up in my default code editor, which for me is Visual Studio Basic. So it's popping open now. And you'll notice that, so I'm going to go ahead and go back to my slideshow for a minute, but we're going to change a few things here. We're going to change the domain to be a little bit smaller so that it runs a bit faster. We're going to reduce the maximum simulation time to 2160 minutes. And we're going to save data every 160 minutes or every 6 hours. So let's scroll down here. And I'm going to go into the section about the domain. And I'm going to change my domain settings to -500 to 500 in X and Y. I'm going to leave the Z alone because remember that 2D is actually a very thin slice of 3D. So I've got the domain set up. I'm going to change the maximum time. I'm going to reduce this down to 2160 minutes so we can actually run a fast simulation in real time here today, not be waiting for a while. And I'm going to change my output interval here and save. I'm going to save the full data every 360 minutes. See anything else? I'll leave the rest out. And okay, so, oh, actually, I got a little bit ahead of myself here. I opened my configuration file, but I already started doing it. I want to walk this back a bit. But notice that when we open up the XML section file, we have several major sections of parameters. We have, the domain settings, which we just kind of walked through, the overall settings of like how long to simulate, time step sizes, things like that. OpenMP settings, like how many threads to use. Here's, in this parallel section. So I'm just going to run 4-threaded for now. How often to save and where, microenvironment parameters, user parameters, and cell definitions. So I'm sorry that I actually jumped a little bit ahead here, but to modify the domain, we go into that domain block. So kind of look over here, and we go to domain, and here's we're going to modify my X and Y bounds to make a smaller simulation domain. So those changes are in red here to show you what to do. And then we're going to modify the save settings. And so I'm going to go over to the overall block and change the maximum simulation time to 2160 minutes. Then I'm going to go over to the save block and say that I want to do my full data save every 360 minutes. So sorry that I jumped right to it here, guys. I was just anxious to go. But yeah, we saved the full data save interval to 360 minutes over here. And let's see here. I should probably check and make sure that there are no frantic questions like, hey, you're not sharing your screen, but it looks like we're in good shape. Okay. And now I'm going to change some user parameters. So I want to change some of the basic scientific parameters of the model. So this is a model of heterogeneity, where every tumor cell has an individual parameter value that says how quickly they proliferate. We're just going to call this an oncoprotein. And so we're going to change the standard deviation to be a much larger value well, actually, we're going to change we're going to leave the mean alone, it looks like, but we're going to change the standard deviation to 3 to allow more variability. So we go down here, and we're going to go past the microenvironment, past the cell definitions, and go down to user parameters. And here we have custom parameters that are really specific to our model. And so we're going to go here. Here's oncoprotein mean, going to leave that alone. Oncoprotein standard deviation, and increase that from the small value here to a much larger value of 3.0. And I'm going to hit, save to save my file every now and again. I'm also going to change the maximum value allowed, where I kind of crop off my distribution to 10. That'd be 1 mean plus 3 standard deviations. So I'm going to change this oncoprotein max to 10. And so now you see that we've opened up a project. We've changed a bunch of the settings in the configuration file. Let's see here. I have a question in the chat. Yes, let's take a pause here and see if there are any questions.
Yes, can I ask a question very quick?
Please do.
My question is, so now you're working on this heterogeneity, I cannot pronounce the project. So you compile it and then you get these settings. My question is, like, this setting configuration is only for this project? When we do like Macklin, and then, for example, like BioRobot, are we going to get different set of like headers and things like that in this settings file?
That's a really terrific question. Yes, that's right. Every project would have its own individual setting file that would be specific to the project. There'd be some elements that are shared, like the domain would be the same. The overall block would be the same across projects. But there was going to be some differences, right? Because every project might have different diffusible substrates. So the overall structure is going to be the same, microenvironment with a bunch of substrates. But the names of them and their values is going to vary based upon the project you're working on. Likewise, the cell definitions will change, right? Because different projects would have different cell definitions. So the overall structure of where you find things will be the same, but what you'll find in there will be different from project to project. And then likewise, that the user parameters, those parameters are very specific to your individual project. So yeah, you're right. You would populate a project, compile, and that configuration file will be very specific to the project. do make reset, and then load another project, and you'll have a different configuration file.
And of course, like one more question in the same
Please.
direction, because let's say, for example, I worked on this project, and let's say, for example, BioWeb project, and then I have like changed lots of parameters and things like that. Then when we do make reset, we are going to lose all of them, right? So how do we, what is the like best practice to keep?
So, and that's a really great question. And I basically see that Mobina and Issy also asked this question. So what is best practice? You're right. You do make reset, and you're going to lose everything you just did. So for the purposes of getting your feet wet and building a project, this is okay. But what you're going to want to do for now is, if you're making a new project that you intend to use in the long term, I encourage you to basically make a new GitHub repository and copy the PhysiCell source into it, and just leave that alone, right? So use your normal PhysiCell for exploration, just playing and learning. But when you're ready to build a project for the long term, make a brand new repository, copy the PhysiCell source into it, make a template, and just kind of go from there. And we can show you later on that you would probably want to modify your makefile, for example, change the name of your executable. Right now, they're not saved somewhere when you load the next project. I think that's something on our radar. And for hackathon people, that would be another possible project, right? Is that we have kind of been prototyping this PhysiCell Studio, but I think one thing that remains is actually like a project format, right? Of how you bundle the source together, and the configuration files and everything else, and save them so you can load and move from project to project without losing these things. And possibly one way to do that would just be to have a way to bundle it in, not in sample projects, but user projects. You could have a very similar style. Package loader like VM, that's getting pretty advanced, but possibly. Yeah, I think a project loader could be a really good addition, and could even be bundled into PhysiCell download, right? We would just have to make a new directory called user projects, and some kind of command to say bundle project or something, or make bundle, and maybe it saves it in the directory. But that I think would be, right now that doesn't exist, but it's definitely a need. Let's go ahead and go back now to the tutorial, but these are wonderful, wonderful questions. And yeah, you're right. I think over time, now that we are evolving from not just a toolkit, but to a full-fledged workflow, we do need an easier way to switch between sample projects and user projects. So I'm excited to see what possible growth will happen there. So what we should do, and probably what I should have done actually while we were talking, is I should have run this thing. So remember, I already did make, and the nice thing about configuration files is you don't have to recompile to load changes in your parameter values. So, I've already built. See how my executable name is at the bottom here. It says it's heterogeneity. So on Windows, you would type heterogeneity and hit enter, and that would execute. But if you're on Linux or Mac OS, you do a / heterogeneity, and that will start running the project. So this should not take very long. And so while it's running, let me kind of show you what's being generated by the project. So I'm going to go into my file browser here. You go, and remember, everything should be spat out into the output directory. So notice that's making a variety of SVG files and XML files and map files. One important one to look at is one called, oops, it moved, legend, where we'll plot a legend of the different cell types in my simulation. Right now, there's one cell type, so it's not terribly exciting, but that will get more interesting later on today. These snapshots are really fast visualization of the simulation as it's going. So that's worth looking at. Oops, okay, I accidentally double-clicked a MATLAB file. But now it's all being generated, and it's making a broad variety of files here. I'm going to go ahead and open up another another terminal while this is running. Okay, and I'm getting out of MATLAB. Whoops. And let's see here. So one rule we can use right away is called make JPEG, and it will convert all of our SVG files into JPEG to get ready to make animations. I think, I should let this continue running. We're almost done here. But it's making a JPEG. There are other rules that are nice too. You can make an animated GIF by typing make GIF, and it will save your result output /out.gif. You type make movie, and it will take those JPEGs that we just made and stitch them together into an MP4 file. So those can be handy for visualization and for sharing your results with others. So this is what's going to be resulting once we're done running our thing here. There is no way to disable terminal output. Thank you. Let's see here. Not right now. So let's see here. Hopefully, okay, we're done running. Let's do make GIF. Just kind of see the animated output. Right now, PhysiCell just sends things to cout. And so there's no easy way right now to disable terminal output, but you're absolutely welcome to ignore it. Down the road, it would be nice to refactor the code to have options to say where you send output from, warning and other error messages. And perhaps we might want to just send that to a text file, for example. So I think that's another thing that could certainly stand to be done. All right, so we're done making our GIF. So let's go into output and look at our animated GIF. So now we can see these different cells proliferating. We see the yellow cells, but the larger parameter values are really proliferating very quickly and quickly kind of take over our tumor. And what I'd like to do now is spend the last, about 30 minutes here just thinking about visualization, and then we can go back and look at more questions. So these are really good questions, I think really point to some growth areas for the code and for the project. So let's talk about loading data in Python. And so we're going to be using a basic Python loader we've written called Python Loader. It's not perhaps the most imaginatively named project, but that's okay. So I'm going to go ahead and just go into GitHub Desktop. And I'm going to go, and I've already cloned it, so I'm just going to check for updates. You see it right here, actually, Python Loader. I'm going to ignore my ignore change. I'm going to ignore my get ignore. And discard changes. And I'm going to pull from origin just to make sure I have the most recent version of code. And so I'm going to assume that you have your PhysiCell directory about the same place where you have your Python Loader directory. But if not, just make sure you have them close together. So I am going to go here. So if you haven't loaded it, you should go here to PhysiCell Tools, Python Loader. Then I would suggest just click on this green button here and click on Open GitHub Desktop. And I already have it, so I don't need to worry about it. But then if you use this to get a few of your codes, hopefully they're close together. So now I have Python Loader. And I am going to show you tricks that you won't need to copy any Python files to your project. So we're just going to assume that I have GitHub/PhysiCell for my PhysiCell stuff, GitHub/Python Loader for the Python Loader. And if you have that relative pathing, this will work out really, really well. And now the other thing is we have a Jupyter notebook for today's session. And so if you go here to the GitHub repository for our class, well, it looks like this is the link to the raw. So we'd have to go File, Save As. And I'm going to just save this thing, not as a text file, but just as a Python notebook in my root directory here for just for today. So hopefully now. Okay, yep, and I can see today's session 2 notebook. So I'm going to go into Jupyter here in just a moment, and into Jupyter Lab, and we'll start using this notebook. So I'm going to go here. I'm just using Anaconda. So I'm going to double click on Anaconda Navigator. Yeah, and Joe points out that you can redirect standard out to a file pretty easily, but that's not the same as silent. No, right now we'd have to figure out some way to redirect that stream to do that. Okay, so I'm going to open up Jupyter Lab. So I'm going to click Launch. And I added all this stuff to my path, so which shall be this, pretty similar. So now I'm opening Jupyter Lab. And I'm going to go into my GitHub directory and see how I have a whole bunch of projects. So this gets back to the earlier question about best practice. I make a new code, let's say a model of like pancreatic cancer. It means I make a new pancreatic model directory, and I copy all the PhysiCell source into that directory, and then I'm ready to kind of go from there. So I use PhysiCell kind of more as my environment for just trying new things. But if I'm going to really work on something long-term, I would make a new repository and copy the code there. All right, so I'm going to go into my PhysiCell directory. And here's the notebook for today. So I am going to open that up. And we'll be able to, oh, okay, looks like I'm going to go here to kernel, and I'm just going to clear all the outputs. So you can kind of start over here. And let's go through some commands. So first of all, we're going to add the directory for the Python loader to our system path in the notebook, so we can load modules and things without having to copy the source all over the place. So we're going to do import sys, and then this command here will open the Python loader, and then we're going to open up the Python data loader here, the whole thing. And of course, the things you almost by reflex always do when you're doing data work in Python, you're going to import NumPy and Matplotlib. And just as a historical note, multicell DS is the name of our, multicellular data standard, and that's why you'll see this MCDS here in the naming. So all this is already into the Jupyter notebook. So if you go to today's source and just download it, you should be able to follow along pretty quickly for this. So this first code block, I'm going to go here and I'm going to hit Shift Enter to execute the code. I'm going to add that system path, and then it is going to import the data loader and Matplotlib and NumPy. And I'm just going to quickly get a list of all the outputs in my output directory. You can see, remember, we saved every 6 hours, so we don't have too many files to worry about here. Let me see if there's a question. Okay, I'm going to leave the discussions to the discussion for now. So yeah, we've loaded up our data loading module, and we're ready to start loading data from our simulation. So now I'm going to, show you kind of how to load a single file. So I'm going to just save this in MCDS, multicell DS. So it's Python MCDS, the name of the output file, and the directory of where you find it. And you can get some basic information like what time did I save, and there's a variety of metadata that's here in this data structure. And so let's just kind of take a look and see what we have. So MCDS, we're going to load that first simulation time. And then we're going to print out some information about the simulation. And if you go to, there are a few things you can do, like MCDS dot, hit tab to kind of see what's there. You see that there are a variety of methods and data. But let's look at the data. And hit dot again, hit tab. And well, there should be some things, but I think what we really want here is data.keys. And we have this dictionary here. We have metadata, we have mesh information, continuum variables, and discrete cells. So let's kind of continue and dig into this and start plotting out some information from our model here. So first thing I'd like to do is to kind of process across all the discrete cells. I'm going to get the variable called oncoprotein. Remember that one that we varied in custom variables, and just kind of see the distribution of those parameter values. And then we'll do some histograms just to kind of plot that and do some quick data visualization. So I'm going to get the mean of all those oncoprotein values, and it's about 1.77. The reason that would happen, right, is because we truncated our Gaussian. So we were supposed to have 1 plus, mean of 1 with standard deviation 3, but we don't want negative values. So we truncated the left half of Gaussian, which means that we skewed the mean to the right. And also there's some selection. Oh, but this is time 0, so no selection yet. Okay, and now let's do some plotting. So I'm going to import Matplotlib. I'm going to hit Qt to do external plots and just kind of set some of the plot size. So here we have a nice little histogram showing the distribution of that oncoprotein. So there are a whole lot of them with a low value, and then kind of a long tail from this Gaussian. Okay, so we can do manipulation of some of the custom data in the simulation. So Arity asked a question if these are readable by peer review. Yes, I think if you contact Randy Hyland with some customization, you can access these files. Basically, the Python loader gives you a way to access the data, but you're welcome to load into any platform you feel happy with. We're just trying to provide you a starting platform. So let's plot the cells. So we're going to go into mcds.data, dictionary key for the discrete cells, and then the discrete cells have a whole lot of things stored in a big table. We're going to get the X positions, the Y positions, and the oncoprotein values, and just save them as CX, CY, and OP. And we're going to do a scatter plot of all the cells colored by this oncoprotein value. And then we'll do a little bit of range work here to kind of truncate the range of the plot. So let's go here into the notebook into section 4. So here we're going to shift enter and get these positions. And so hopefully this notebook is like a nice reference material for you to modify for your own visualization. And now I'm going to do the scatter plot in this external window. And you notice that this thing is like really, really ugly. So we need to do some things to improve our plot. So we'll get into that in just a moment. But we do have this nice scatter plot of easily accessing some parameter of the different individual cells and plotting XY positions. So for not much work, we have some visualization, and it should give us a lot of capabilities, actually. So now what we're going to do is, first of all, I'm going to make the plots use a bigger size for the cell dots. So I'm going to change this. Now, this is going to vary a lot by the resolution of your screen, so you may need to play with this parameter value. So I'm going to do the X positions, Y positions, the coloring based upon the oncoprotein, and then the size of the points. And then I'm going to get rid of this ugly, where I stretched out the plot, and make sure that it plots with the correct aspect ratio, and then redo the axis. And then we'll add a nice color bar on the end and add some labels. So kind of the beginnings of a better plot. So shift enter. And now my plot, see, now these circles are circles, and we have the tumor plotted nicely. We may want to play more with the dot size a bit, but it's, again, it's a start. And so we can do a lot to start customizing it. What I'd like to do, though, is show you how to load a different time. So we're going to go and do the exact same stuff, basically. We're going to load a different simulation time, kind of display the information. We're going to get the X and Y positions again in the oncoprotein and see how the histogram has changed. So let's go to section 6 in our Jupyter notebook, hit shift enter, kind of run those commands. And let's see here. Here is my plot. And see now that the oncoproteins have changed, right? We see this shift towards the right because the faster growing cells are being selected for. So they are starting to dominate the histogram. So we can see, how changes in our data as we load different times. Now, the nice thing about Python, of course, is that you can do searches, and you can look for cells and kind of sub-select them and plot only parts of the cells. So we're going to, each cell has a cell cycle with a unique code. Any code that's greater than 100 is a dead cell. And so we can use that to actually search for the live and dead cells. So I'm going to do cycle. So I'm going to get this variable of the cycle to make it easier to access. And then I'm going to do as type to recast it to integers. And I'm going to use argwhere to find the cells that either are above 100 on cycle, so they're dead, or below 100, which means that they're alive. And then I'm going to flatten this to make it a nice flat vector. So let's go to section 7 to perform this nice little search. And we can see, by printing the length of these lists, I have about 2,000 live cells and about 90 dead cells. So we can do searches very nicely. And then we can, get the counts like we just showed you, and get the mean oncoprotein only live cells. So let's see here. I think this is in section 8. So yeah, we can hit section 8, start getting some information, changing our scatter plot. So now I'm only plotting the live cells, right? So you see how we can use these sublists, the indices, to really do nice restricted plots, because I'm plotting CX only on the live cells, Y position only on the Y cells, oncoprotein only on live cells. And here I just, for the fun of it, did some smaller dots. And of course, changed all of your title and everything. So using these kinds of searches, using argware, you can really get some more detailed and nuanced sub-selections of your cells to visualize and analyze. Okay, so let's do a little bit more data loading. For example, let's do a fancier search. Let's look for all live cells with an oncoprotein value greater than 5. So you can do this argware, you can actually do ampersands, you can do and operations, say, find anything where my cycle is less than 100, and if the oncoprotein is greater than 5, and then flatten that list and do a scatter plot. So this is in the next section of our Jupyter notebook. And let's see here, what do we got? Here, yeah, see, we have live cells with protein greater than 5, and that they're also alive. So that's kind of nice. You can really do some restricted researches if you want to do some more sophisticated analysis. So that was in section 8A of the code of the Jupyter notebook. And now I want to talk a little bit about doing substrate plots and contour plots. So the basic idea is you get you can get the list of the substrate names. And here we want to do get the concentration of oxygen and then generate a mesh and then do a contour plot. So here we're going to see, okay, in this particular simulation, we've only got one diffusing substrate called oxygen. We can call this command here to store oxygen in a variable, generate an XY mesh for contour plotting, and then do a filled contour plot. And we get a nice oxygen plot over here to go with our simulation. And of course, you may be interested in overlaying these. So now we can plot oxygen on the background and maybe with a different coloring map, for example, and then do a scatter plot of the cells on top of that. And so that's what we're doing here. So we're just kind of, stringing more and more commands together. And so if we look, now we have not the most attractive plot, to be honest, but we have a plot of the oxygen with a contour bar, a color bar over here, and then the cells colored according to their oncoprotein on the top of it. So you can do quite a lot in this. You can also, of course, loop this to do time series analysis. So here I'm going to say, I'm going to do a range, load the appropriate file name. So here I'm just kind of doing some systematic naming the files to access the different files. And then I'm going to get histograms and plot them and get all these different plots. So it's kind of, you might want to browse this, I don't want to run out of time here. So, but notice here, we're loading the file, creating the file name, loading the file, adding the current time to the list of times as a variable. Then we get like the live and dead counts and the mean and standard deviation of these parameters and kind of plot them out over time. So I'm just kind of looping it out over time. And let's do something with that. So I'm going to plot the live and dead cell counts over time. I'm going to do a polynomial fit to the logarithm to estimate the birth and death rates. Okay, so here first I'm going to plot. So I have my live cell curve and my dead cell curve. And now I'm going to do a polynomial fit to the live count. And so that would be an estimate of my exponential birth rate for live cells, live cell population. And of course, we can do other plots, like plotting the mean, for example, as a function of time. Notice that the mean of the oncoprotein is going up, which shows that we're getting some kind of a selection process that is not keeping the same distribution. Okay, so now we're kind of done running through one project. So I'm going to do make data cleanup. So what that does is it removes all the simulation data that we just created in the output directory. So if that would get us ready to run another run. Of course, as you learn, right, if you go into the configuration file, you can change the name of your output directory, which means you can make output 1, output 2, output 3. And as long as you remember to make those directories, so they have someplace to do the writing, then you'd be able to save different simulations of different subdirectories, and that might be helpful. So now we're just going to really quickly return to a clean slate, make reset. And I want to just show you a little bit more visualization on an example where we have multiple cell types. And so I'm going to do make data cleanup, make reset, just did that. So first I cleaned up my simulation data, and then I returned to a blank slate. So I'm ready to populate another sample project. And so the sample project we're going to model here is use the biorobots sample. So I'm going to type make list projects. Actually, let's do it over here. Wider window. And so we see all these different projects that are available. So make biorobots sample. And I'm going to edit the configuration file. Now remember, I already have the configuration file open in my code editor. And since I changed projects, it should now open up the new one and kind of refresh. Yeah, so notice that this is all different. Serhi asks if we have a nice way to deal with many large output files. Not at the moment. We could do in other file formats, but we'd have to write that format and make sure it's cross-platform compatible and self-contained across all platforms. So that would be one nice way to do it. But that's a possible project. Okay, so what we want to do is we want to modify this example to run much less time, to only 720 minutes. So I'm going to go over here to the overall, because we want to just quickly generate some simulation data so we can do some more visualization. I'm going to do a full data save every 240 minutes. And I want to save the SVGs every 30 minutes rather than every 2 minutes. So there'll be a lot less data here. And that should be everything. Oh, we need to, of course, remember to compile the project. So we populated it, but we have to type make to compile it. Now, because of a lot of our parts have already been compiled, this should be pretty fast. It should only be compiling the things that are new for this project. The name of the executable is biorobots. So we'll type that and let it run. This should be a really fast simulation since we only need to simulate 720 minutes. And there's not a lot of proliferation going on in this example. Okay, so yeah, we're already like 1/3 done. Okay, so we're running. Oh, yeah, let's take a look at the outputs here, by the way. So I'm going to go back here and look. Let's look for that legend file. So notice here now, the legend is more interesting because we have multiple cell types. We have default, which is not used in this particular project, director cells, cargo cells, and worker cells. So we won't worry too much about what that simulation is doing, but we want to be able to plot these different cell types. Okay, and looks like we've reached the end of the simulation. So let's go back to the notebook and we're going to load the last time. So let's go here into our Jupyter notebook. And I'm going to load the very last time. And now let's get ready to do some plotting. So just kind of remember, we construct our file name for a particular time. We load the data file, we get the current time. And I'm going to save 3 indices. I'm going to save index 1 be the list of all cell types of type 1, which are my director cells. Index 2 is a list of all cells of type 2. And 3 is the indices of all cells of type 3, which will make it easier for us to do these plots. And then I'm going to do a scatter plot. So I'm going to do I'm going to do a scatter plot of the type 1 cells, going to make them green. Type 2 cells, make them blue. Type 3 cells and make them red. So let's see, I'm restricting my X positions to these different sets of indices. I'll just get a little bit more precise handling. So here in section 14, I'll go ahead and do that in our Jupyter notebook. And yeah, see our plots now. Although right now I might need to change my circle size a little bit. So maybe I'll just resize my window for now. But we can see these different cell types being plotted with the different colorings. And the rest we can do just mostly to start prettying it up. And of course, we can also overlay this on a contour plot of the cargo signal. So I'm going to get the list of substrate names, and I'm going to, out of all those, look for the cargo signal, which is released by these blue cells, and just overlay the 2 plots. So we'll kind of do that. And yeah, so we have a contour plot. It's not much to look at in this particular one because I did like gray. Maybe we should just do, I don't know. I can never remember the name of I think I'm just going to get rid of this. Just kind of do whatever their default color map is. And of course, we can turn on things like a color bar. And so, we can add quite a lot here to our plots, but we've kind of gone through the basics now of how we can populate a project, compile it, modify the parameter values, and then run a simulation and start loading the data into Jupyter to visualize things like individual cell properties, their positions, and kind of change and search for subpopulations of cells based upon their properties. And then we can combine them with contour plots of the substrates. And so let's kind of go back here. And that's really, where we need to be. So we have the really basics, right? We can take an existing project, modify it and run it. But of course, what you really want to do is make your own projects where you have, I think, a deeper understanding of what you put into them. So in the next few sessions, let's see, let's close this one. So I'm going to just kind of clean up, data cleanup. So I'm back to a clean slate. So I'm ready for later on today. But this now depopulates all the custom stuff and goes back to the kind of the default clean running stock. And by session 4, you'll start working on the intermediate workflow, where we're going to make a new model rather than an existing model, starting with a template. We're going to plan out what belongs in the model, what diffusing substrates, what agent types. You're going to populate and build a template project, and you're going to open up the model builder GUI, start editing your domain, your microenvironment, your cell definitions, then run and view the results. And I really liked the questions we got here today. I think that is a need to be able to have a way to very easily save a project in a subdirectory of user projects, so that you can easily switch between the samples and your own projects without worrying about losing things. So hold that thought. I think it'd be a really good idea. So we have actually, amazingly, we have finished a little bit faster than we meant to go. Sorry about the pace if it is too fast, but on the flip side, we do have some moment for some questions and to start thinking about the next session that we'll start up after our break. Are there any questions? And thank you, by the way.
I may have sorry, I have one question regarding because I was trying to follow up your workflow, and I noticed one thing, that the simulations and the results from simulations are different between rounds, even though we had same seed number in the config file.
That's a really good question. Unfortunately, there is some stochasticity in the run order when you multi-thread, and there is no guarantee of thread execution order, at least not without some significant work. And so even if we have the same seed, if we have the different number of threads, or even same number of threads for different architectures, there could be some variation on which threads run first, and that can lead to some variation in the downstream output. From a computer science perspective, this is not great, right? Because you would like an individual run to be reproducible with the same seed. Scientifically, it's not as bad, because for a stochastic model, you're never going to run just one replicate anyway. You're going to run a whole bunch of them and then look at the emerging statistics. And that tends to be reproducible. But it is a really good point to note that what you see and what I see can be different.
Okay, so if I run the project again with the same setup, if I run the project in the same setup on the same machine, it should reproduce exactly.
I think so, but again, when it comes down to your operating system scheduling, there's still no guarantee that you have 1,000 cells divided between 4 threads and they will all execute in exactly the same order. You might have a virus scan running in the background, or an email or something that slows down one thread or another thread, and they run at slightly different pace. So I wouldn't make that promise, probably.
Okay, thanks.
Yeah, Randy Highland has done some work on getting down to bit level reproducibility, but ultimately, if you want single run reproducibility right now, probably the safest thing to do is to run single threaded, which might be good if you're debugging. But it's a really good point that you will see some variation. Other questions? I'm seeing some comments in the chat, which is really good. Julio asked a good question. Is there documentation available for PhysiCell and Python loader? So both of these are really great questions. And this has unfortunately been the problem of being a small lab, is we have not been able to keep up with the documentation as well as we'd like. Basically, the workshop from 2021 and now this workshop are our primary documentation for PhysiCell. I'd say the primary documentation for the data loader is the slides in this talk. They definitely are a need to expand the documentation. We originally had a PDF user manual, but it was incredibly unwieldy to update. And so I think one of our big to-dos, the thing I plan to start on in the fall, is to start building a true, either probably just GitHub wiki pages, in Markdown, and to build a user manual based on Markdown, because I think it'll be much, much more maintainable. But clean documentation that keeps pace with development is a really clear need for the community right now, and something that I think we need to spend some more time on. So I really apologize for that. I think after about version 1.4 or 1.5 or so, we stopped keeping pace on the user manual, and the primary documentation now is in the tutorials from last year's workshop, and then the tutorials we're generating for this workshop now with you live. Doxygen. I hate Doxygen. Let me give a better answer to that. Doxygen requires so much work mucking with the files and generates quasi-human readable output that I haven't found it hugely productive for educating humans on how to use PhysiCell. But I would be very willing to do a user wiki. I think that'd be a great idea. So thanks for those points. We looked at Doxygen like 10 years ago, and it was just so much work to insert very structured comments into code. It requires so much refactoring that it doesn't seem like a fruitful way to go, but I would love to be surprised on that. And then Randy points out that there is some documentation on using Python loader also on our blog. Thank you for reminding me on that, Randy. So do look on that link. That there are some full run-throughs on how to use Python data loader here on our blog, and I think one thing we could certainly stand to do is probably nuke and start over on PhysiCell.org and reorganize all these materials with better links so people can actually find them. So there is a real weakness right now. I think that things are pretty scattered and definitely an area of need. Other questions? These are really great questions. Yeah, Mubina, please.
Yes, I wanted to ask, what is the base of different time scales for different setting files?
That's a really great question. I'm going to go back into the parameter file just to easily show those. Notice that there are these different time scales here for diffusion, mechanics, and phenotype, or deep. And basically, because the biology is so multi-scale in time and space, that we don't run every process at the same scale, which means that we don't have to run everything at every time step. And so after a lot of analysis and convergence and accuracy testing in particular for the underlying diffusion solver, we found that for typical diffusing substrates at the scales that we care about, we found that for our solver, a time step size of 0.101 minutes was sufficient to maintain accuracy and find that balance between accuracy and performance on diffusion. Whereas mechanics, we can get away with a slower time step size of 0.1 minutes because cells tend to be moving on the order of microns per minute. And then for phenotype, that's for resolving things like cell volume changes or division, which tend to happen on hours-long time scales. And so we use a step size of 6 minutes for that by default. You can change these. But we found these parameters tend to work pretty well. Now, if you're going to go into microbiology, where the biology is a lot faster, you may want a smaller phenotype time step size, because if you have cells cycling every 180 minutes, maybe you might want to resolve that more. Or if the cells are moving faster, you might want a smaller mechanic step size. But that's the origin of those different step sizes. It's a really good question.
And between different, I checked for tumor cells and stuff, and they were different with these. For each of them, you have your considerations for different time scales.
I'm not sure if I understand the question, sorry.
There were biorobots, and there were some different kind of cells.
Okay. Getting down to the parameters of the cell definitions. Rather
yes,
than
and
right?
They were different with each other in different settings.
Okay, well, the step sizes are probably the same, but the time scales of like proliferation and death would probably vary from problem to problem based upon what you're trying to model. Yeah.
And I wanted to ask, what are you integrating your solving Navier-Stokes?
Right now, Navier-Stokes is not on our radar because that tends to happen at very, very fast time scales compared to what we're doing. And you would spend all your time on Navier-Stokes. But that said, if someone has an interest, I'd be very interested to talk about that.
Mm.
So right now we are kind of at that diffusion time scale, which is
Yes.
much, much slower compared to advection, of course. We have
Yes.
talked with Alison Marston's group in particular to integrate or couple with SimVascular, and that might be an option for starting. But I think otherwise, we may want to think about, integrating a full-on, finite element code if we want to do Navier-Stokes. And probably the easiest one to couple would probably be FE Bio, I think. That's a really good question.
Okay. Thank you very much.
Thank you. Well, I think here, let's not go too much over time. We want to make sure you have time for a break. According to my agenda here, we are on break until 1 o'clock US Eastern time. I'd encourage members of the hackathon to go back together to the virtual fountain, kind of continue meeting your colleagues. And then we will start right here in the virtual lecture room in this same Zoom link at 1 o'clock US Eastern time, which is in about 45 minutes. So thank you very much, everyone, for dialing in today. We hope to see you throughout the week in our virtual workshop. And we look forward to continuing. And thanks for all the wonderful questions. And we will post a copy of this recording on YouTube later this evening once we're done processing the video. So thank you very much, everyone. And we'll see you really soon.